Well, I want to welcome everyone to today's RNA Collaborative Seminar Series. And I should point out that this is sponsored by our wonderful RNA Society. For those of you who don't know, I'm Lynne Maquat. And speaking of the RNA Society, I want to tell you two things before I introduce our speakers for today. And the first is to remind you that there is a RNA Lightning Talk workshop and competition that is sponsored by the RNA Society. This is open to all trainees, and the deadline for submissions is March 1st. You will see the details of how to apply in the chat link that Liz is going to make available to you. There will be cash prizes for first through third place at a workshop event that will occur on April 27th. And then I also want to remind you about the next RNA Collaborative Talk, which will happen on Wednesday, March 9th at 10 a.m. Eastern Standard Time. This is hosted by the EMBL, the European Molecular Biology Lab, and the speakers will come from the AFRUSI and the HENEG group. So with those two announcements, I am going to go ahead and introduce our speakers. Our first speaker is Peng Yao. He is an associate professor in the Department of Medicine in the Cardiovascular Research Institute. He's also an associate professor in the Department of Biochemistry and Biophysics and a member of our University of Rochester Center for RNA Biology. And he's going to talk about hyperactive translation programs during cardiac remodeling. So Peng, go ahead and show us your slides. Talk for about 25 minutes. We will have 5 minutes of Q&A, so you can introduce the questions you have using the Q&A option at the bottom of your screen. Thanks, Peng.
Yeah, thank you, Lynn, for your kind introduction. So hi, everyone. So today I'm going to talk about hyperactive translation programs during cardiac remodeling. My lab is working on understanding translational control mechanism involving heart disease. So as you all know, the translation is the last step of central dogma that produce proteins that perform all kinds of cellular activities to prevent human disease and maintain homeostasis in physiopathology. So as we know, the overproduction of pathogenic protein or insufficient expression of protective protein could drive human disease, such as heart disease. There are recent study showing that in human hearts, there's a lot of mRNA undergo translational dysregulation, but mRNA steady state level stay unchanged. So we are trying to fill the gap to understand the translation control mechanism behind all of these observational studies. And by asking three major questions, what is the impact of translational control in heart physics and pathology, and what are the molecular mechanisms of translation regulation in cardiac biology? And by answering the first two questions, hopefully we can provide implication of translation regulation in heart disease treatment. We generally start with data mining of unbiased screen, identifying translation factors involved in cardiovascular disease. By using genetic and disease animal model plus pharmacological inhibitor, we can perform multi-omic study to determine molecular mechanism and discover novel therapeutic target. Here's an overview of translation machinery in mammalian cells. There are four major components, such as amyloidsal tRNA synthetase, like EPRS here, that provide proleo tRNA proline as a building block for protein synthesis. And also there are a bunch of translation factors, such as elucidation factor EF4G1 and EF4G2, that can assemble the ribosome for protein synthesis. And also there are many translation regulatory factors, such as RNA binding protein, like the RNA helicase DDX3X that unwind the structure, and the microRNA that binds to UTR region and regulate translation efficiency. So today I'm going to talk about two stories. One is focusing on EPRS and EF4G2 that promote pro-fibrotic mRNA translation in cardiac fibroblast. And the second story is about double-stranded RNA element that can regulate URF MAOF translation balance in cardiomyocytes. So the first story is mainly about a proleo tRNA synthetase and the amyloidsal tRNA synthetase that can regulate proline-rich protein synthesis in cardiac fibrosis. So in general, in normal healthy hearts, there are two major cell types, cardiomyocytes and the cardiac fibroblasts. So the cardiomyocyte is a major pumping unit cell type for pumping the blood out of the heart to provide oxygen and nutrition to other part of human body. And the cardiofibroblasts can synthesize and secrete base level of extracellular matrix proteins, such as collagens, to stabilize the heart. But when the heart undergo cardiac injury or stress conditions, such as hypertension and myocardial infarction, what happens is the cardiomyocyte will undergo hypertrophic growth, and the cardiofibroblasts will become activated in your myofibroblasts that can dump out a huge amount of collagen and other ECM proteins that cause the stiffness of myocardium and leads to heart failure or arrhythmia and sudden death. For a long time, it has been well understood that TGF-beta signaling and the beta-adrenergic signaling can activate mRNA transcription of the ECM genes and also can activate translation of those proteins during synthesis. But however, the translation activation mechanism is still unknown. We believe it possibly can be enhanced at both elongation and initiation steps. If we simply look at a collagen molecule, and we can see there's a large number of protein-rich codon motifs like XPPY, the PP dipeptidyl motif surrounded by any amino acid. And if we look at a typical collagen 1 protein, you can appreciate there are so many PPG motifs throughout the protein. And then the prolyl tRNA synthetase, or PRS, is essential for catalyzing the proline ligation to tRNA proline, allowing the decoding of 4 proline genetic codons. Combining with the EF5A elongation factor, EPRS is essential for translating proline-rich mRNAs, which encodes profibrotic proteins. So the first thing we look at is using the patient-derived cardiofibroblast treated by TGF-beta versus vehicle, followed by RNA deep sequencing data analysis. We found among all translation factors, glycotRNA synthetase and EPRS among the top 3 most induced translation factors. And we also confirm in our own hands that EPRS mRNA and the protein level are induced in failing hearts compared to long-failing human hearts. So the idea is that we can hypothesize that cardiac stress can induce EPRS that promotes fibrosis via increased prolyl tRNA proline pool and translationally activate profibrotic proline codon enriched mRNA in cardiac fibroblasts. If we can reduce EPRS expression or inhibit enzymatic activity, we hopefully can prevent the translation of proline-rich profibrotic mRNA and also compromise fibrosis without affecting global mRNA translation. To proof of the principle, we use CRISPR to generate the EPRS heterozygote knockout and subject the mice to transverse aortic constriction surgery that mimics human hypertension. We found actually the knockout animal heart is very resistant to cardiac fibrosis, indicated by lower collagen deposition. We also confirmed the same phenomena using a conditional inducible knockout in the myofibroblast cell type specific manner using peroxystein-Cree recombinase. 14 days post the TAC surgery, we still see the cardiac fibrosis is dramatically reduced, suggesting that EPRS haploinsufficiency can actually resist cardiac fibrosis. But however, we cannot make such genetic manipulation in human patients. So we need to find an EPR specific inhibitor. And luckily we have halofuginol or halo that has been used for thousands of years in Chinese medicine, Changshan, to treat malaria infection as well as cardiac fibrosis disease, arrhythmia. And most recently, crystal structure data suggests that halo can actually bind to PRS catalytic domain, blocking the proline ligation to tRNA proline. So we use the same compound halo to treat the TAC surgery model. We found there is a significant reduction of cardiac fibrosis. And then we further look into the downstream molecular mechanism or targets of EPRS by treating mouse fibroblasts with low dose of halofuginol at 100 nanomolar, followed by RNA sequencing, polysomal sequencing. Well, we found majority of the mRNA that are enriched in area 4, in area 1, both mRNA and the translation efficiency level are reduced. And however, in area 3, both mRNA translation efficiency are induced as a compensator response. If we look at the gene ontology analysis, the top 1 most enriched pathway is ECM receptor interaction pathway. In the downregulated area 1, including all of the collagen coding genes, and they contain proline-rich codons. And in area 3, we found most of the induced genes actually are encoding ribosomes, proteins, translation factors, tRNA synthase as a compensatory response to fight against the proline starvation by halofuginol treatment. And interestingly, EF5A and GDBP2 are 2 ribosome stalling resolving factors that is significantly induced, suggesting that there's a ribosome stalling at the proline codons. Then we overlap the proline dipeptide motif in enriched genes through human and mouse genome, together with our own RNA sequencing and polysome sequencing indicated downregulated genes, and also quantitatively mass spectrometry analyzed the proteins in human cardiofibroblasts treated with halofuginol. We found there are 83 common hits, including majority of them are extracellular matrix protein, including most of the profibrotic collagens and also ligand receptors and enzymes. However, long proline-rich genes, they are not translationally silenced by halo at all. And one of them is a sulfatase enzyme called sulfa-1 is found by us as a novel biomarker for cardiac fibrosis. And very excitingly, just a few months ago, there's a lab showing a paper in bioRxiv by screening a large number of FDA-approved compounds, and it shows that halofuginol is the top 1 most potent inhibitor of SARS-CoV-2 infection. And the mechanism and how it works is actually by blocking the translation of both viral proteins and the host receptor proteins that contain proline-rich codons, such as the heparin sulfate proteins like SALF1, as we have discovered, collagens, as well as the most important ACE2 SARS-CoV-2 receptors. So hopefully I convince you that EPRS is a driver translation factor for cardiac fibrosis, and the EPRS knockout mice display resistance to cardiac fibrosis, and the halo can reduce fibrosis in mouse heart failure models. And we have discovered 83 proline-rich profibrotic target mRNA of EPRS, and one of them, sulfa-1, is a novel cardiac fibrosis biomarker and anti-fibrosis target gene. So now we know EPRS is required for translationally elongate on the proline-rich mRNAs, but how about at a translation initiation level, whether ICM mRNA are enhanced during the initiation step? And to make the long story short, we found actually initiation factor 4G2 or EF4G2 is involved. So what is EF4G2? Dr. Shinya Yamanaka over 20 years back have shown that EF4G2 is required for embryonic development and also embryonic stem cell differentiation. And for the last 2 decades, EF4G2 has been shown to activate translation of specific mRNA under stress condition, such as ER stress. And most recently, people like cardiologists, they realized that EF4G2 is induced in dilated cardiomyopathy heart failure patients' whole heart, and it also is induced by TGF-beta in primary human cardiofibroblast. So we confirm in our hands that EF4G2 is significantly induced in dilated cardiomyopathy heart failure patients, cardiofibroblast, compared to long failing hearts. And by locking down EF4G2 using lentiviral shRNA, followed by RNA-seq and polysome sequencing, we found majority of the mRNA translationally downregulated, but the mRNA level doesn't change that much. And among the translational dysregulated genes, a lot of them are actually coding collagens or ECM proteins. If you look at the gene ontology terms, the most top 3 downregulated at a translational level actually are the ECM receptor interaction pathway, including all the profibrotic collagens. And very interestingly, if you look at the compensatory response, which are the translationally upregulated genes, there are some anti-fibrotic collagen, like collagen 5A3, are actually included, going to the same direction with the reduction of the profibrotic collagens. So we believe that possibly knockdown of EF4G2 can reduce cardiofibrosis. To prove this, we knock out EF4G2 in myofibroblasts using an isoproterenol injection neurohormonal heart failure model. We did find cardiofibrosis dramatically reduced. So for the sake of time, I'm summarizing some of our major findings here. We found the EF4G2 is induced in the heart failure human hearts and also myocardial infarction mouse heart in vivo. TGF-beta can activate EF42 protein expression that will boost up ECM-related mRNA translation like collagens. And if we knock down EF42 in cells, we can reduce total secreted collagen, preventing migration and proliferation of cardiofibroblasts, and also block the cardiofibroblasts to myofibroblast activation in vitro. If we knock out EF42 in vivo in a mouse model, we can get a much less fibrotic heart and also partially preserved cardiac function. So now we know that cardiac fibrosis is a major hallmark for heart failure, but there's another hallmark for heart failure as well, is the cardiomyocyte hypertrophy growth, which will lead to myocyte apoptosis that leads to heart failure. And generally, translation is dramatically enhanced during cardiomyocyte hypertrophy, but we still don't understand how the translation program is actually enhanced. So as we know, there are many RNA elements localized in 5' on-trans region, including upstream operating frame that I highlighted here. Once it is translated, it can inhibit main operating frame translation. And also there are stem loops or double-strand RNA structure that can activate or inhibit translation depending on where they are localized and how strong the base pairing are. And to make long story short again, our finding is that the double-strand RNA that is localized downstream of the AUG start codon within a URF actually can activate URF translation. And as a consequence, it can inhibit the main operating translation. So the central question we're asking here is, could 5'-UTR, URF, and double-stranded RNA elements can play a synergistic role in regulating mRNA translation in cardiomyocytes? So to answer this question, we data mine ribosome profiling databases from both human and the mouse heart failure samples. And we did find a lot of the URF are occupied by translating ribosomes, producing micropeptides that have been validated by mass spectrometry analysis. And the top two most enriched pathways are actually transcription factors, including a number of cardiac transcription factors such as GATA4 that encode small peptide. The protein sequence is not conserved. However, the URF is very, very conserved. So then we predict their structure. We found actually there is a very common feature like the downstream double-strand RNA structure of the AUG start codon within URF across different cardiac transcription factors. And we choose GATA4 to further focus on is because GATA4 is a key transcription factor required for cardiomyocyte growth. It is known that GATA4 protein level is significantly induced in human heart failure and the mouse heart failure models. And also GATA4 can drive cardiomyocyte hypertrophic growth since it can activate transcription of a bunch of pro-hypertrophic gene expression. So the first question we're asking here is, what is the role of URF in regulating GATA4 expression? And I believe this is also a chicken egg question. How are the transcription factor encoding mRNA regulated during their translation in the cytosol before they can enter the nucleus to bind DNA and control mRNA transcription? And to answer this question, we basically use a human ESC to cardiomyocyte differentiation model. Basically, we can generate a beating cardiomyocyte in the Petri dish. And then we use CRISPR-Cas9 to knock in a single nucleotide mutation from ATG to TDG. And then we found in the homozygote knock-in cardiomyocytes, the GATA4 protein is induced by 2.2-fold, suggesting that basically URF can inhibit the GATA4 main RNF translation and limit the gene expression. And more interestingly, we found there are more cardiomyocyte differentiated from ESC, and also the cell size is more enlarged, suggesting there is a spontaneous cardiac myocyte hypertrophy. So this really confirmed that the GATA4 URF can inhibit main operating frame translation and prevent spontaneous cardiomyocyte hypertrophy. So then the next question becomes, how is this URF and main RF translational balance being regulated? We know there are INSYS in-trans mechanism. I only have time to talk about the INSYS mechanism here, which is mediated by a double-strand RNA element, as I mentioned, that is downstream of the URF start codon acting as a translational activating element of URF. So we first use the shape assay to confirm that downstream of the AUG start codon, there's a double-strand RNA conformation. If we mutate CA to AC to disrupt the two base pairs in this dsRNA element, we basically can unwind this secondary structure to form a single-stranded structure. And then we also introduce the same structural mutation into a dual luciferase reporter. Compared to the wild-type reporter, there is an increased translation of a main operating frame of the luciferase. If we rescue these structural defects by swapping the GU to UG on the other strand, and basically we can regain the inhibition of the main operating frame. As a part of control, the ATG to TDG URF inactivating mutation has a constitutive activation of the main operating frame. So the mechanism we believe that is happening here is there's a downstream double-stranded RNA that actually can store the 40S pre-initiation complex, waiting for the 60S to join to form ADS that can translate this URF small peptide and drop off at stop codon without translating the main operating frame, that is GATA4, so which will help to reduce GATA4 level and limit cardiac hypertrophy. On the other hand, we also have data to support that DDX3X as RNA helicase can unwind this double-stranded RNA, allowing the pre-initiating complex to bypass the URF and recognize the main operating frame and translate more GATA4 protein that will drive cardiac hypertrophy in a different direction. So now we understand that URF and the main URF has a seesaw relationship between each other. If there are more URF being translated, there will be less main URF translated. So then we come up with a general idea by using antisense oligonucleotides to fine-tune protein expression to maintain homeostasis and prevent disease. So basically, we can design ASO either to enhance URF activity or inhibit it in order to either reduce pathogenic protein expression or restore the protective protein expression to actually prevent disease. So here, I'm showing you, we designed two types of ASO, ASO1 versus ASO2. So ASO1 binds to the upstream strand, unwind this double-strand RNA element. ASO2 did the opposite. It can base pair with the downstream strand, which can form even a more stable double-strand RNA. So we use shape assay to confirm that ASO1 did unwind the secondary structure, and then the ASO2 actually forms even longer and a more stable double-stranded conformation by base pairing to their target strand. And then to test these ASOs, we first use ASO1 to treat the same dual luciferase reporter system, and we found ASO1 can dose-dependently induce the main operating frame translation by bypassing the pre-initiating complex through the URF, and then the ASO2 can dose-dependently decrease the translation of main operating frame. And then we also confirm in AC16 human cardiomyocyte, the ASO1 can dose-dependently induce the endogenous GATA4 protein expression, and the ASO2 can decrease it. So, but we still don't know whether it works in vivo. So that's why we use nanoparticle to deliver the ASO2 that inhibits the GATA4 main operating frame translation, and then inject them into the isoproterenol-induced heart failure mouse model. With just 2 doses, we found actually that with the control ASO treatment, the isoproterenol can induce the cardiac hypertrophy, but the GATA4 ASO2 significantly normalize the heart size. And this is quantified by also the cardiomyocyte cell size that is dramatically reduced by the GATA4 ASO2. As we expected, the GATA4 protein expression is significantly reduced by the GATA4 specific ASO2. So as a summary, I hope that we can convince you that the GATA4 URF can inhibit GATA4 main operating frame translation and limits cardiomyocyte hypertrophy, and that the double-strand RNA element can complement with the weak Kozak sequence contacts to activate URF and inhibit main operating frame translation. And the RNA helicase DDX3X can unwind this double-strand RNA element and modulate GATA4, URF, and the main URF translational balance. And then the URF exposing and sequestering ASO can either promote or inhibit GATA4 main operating frame translation in the cardiomyocytes. And by utilizing the URF sequestering ASO, we can antagonize hypertrophy in both human cardiomyocyte as well as in the mouse cardiomyopathy models in vivo as a potential therapeutic approach. So in future, as a take-home message here, that we believe that hyperactive translation that is happening in both cardiomyocyte and the fibroblasts will be a key etiology of heart disease based on the three major discoveries that we have made here. And then finally, I want to thank the members that contributes to this work, and also thank for CVRI faculty and RNA Center faculty for their collaborations, and also I appreciate for the funding resource. So thank you so much for your attention. I'm happy to take any questions. Thank you.
Thank you, Peng. That was excellent. So I don't see anything in the Q&A box yet. If you have questions, we have about 3 minutes, and I'll start. So Peng, that was really excellent. I like the way you've tied together your two stories. You discussed the mRNAs that are translationally dependent upon EI or upregulated by EIF4G2, but not the features of the mRNAs that allow it to be recognized by this 4G variant. Can you just remind us what they are?
So you mean the RNA cis-acting elements that possibly can be recognized by EIF4G2? Yeah, that's a great question. So we are trying to do some RIP sequencing assay, and then we have some preliminary data suggesting that some of the mRNA that actually are directly bound by EIF4G2, but we try prediction to see if there's a IRES, like internal ribosome entry site. Elements are there. Some genes that contain putative IRES, but some do not, suggesting that the IRES may not be the common feature for the EIF4G2 specific target mRNA. And we are trying to figure out whether there are other structural or sequence specific features on the 5' UTR of EIF4G2 target mRNAs in the future. And hopefully we can further understand. That's a great question.
So you have two questions, and then we'll have to move on. One, very interesting talk, Peng. Curious about the 5' UTR ASO that enhances translation. So ASOs don't block 4DS scanning. That's the question.
Mm-hmm. That's a great question. So what we believe is the ASO actually binds to the downstream pass strand that is after the AUG start codon, but it's not strong enough to resist the ADS unwinding activity. So the ribosome, once fully matured or assembled, it is one of the most strongest helicase in vivo. So we think that this ASO binding to the target strand actually function as a roadblock for the pre-initiation complex, the 4DS, but it does not prevent the ADS from unwinding it. So the 4DS will store at the AUG codon and recognize it, and then get assembled with 6DS, but then ADS will basically unwind the ASO and then continue the translation.
Okay, and another question, is the function of the double-stranded RNA motif dependent on the sequence? Did you test more variants of the double-stranded region?
Yeah, that's also a very interesting question. So we believe that when we compare different cardiac transcription factor mRNA, looking at the downstream double-strand RNA, there's no homologous sequence feature among them, suggests that it is unlikely it's dependent on the sequence of those double-strand RNA, but rather than there is a structural feature that is very commonly observed in those potential cardiac transcription factor mRNA 5' UTR. So we believe mainly is dependent on the structural feature rather than the sequence.
Right. And I have to take Bill Marsleff's question. He's asking, and the answer is yes, but I don't think you know what it is, but defines the subset of mRNAs that are recognized by EIF4G2, similar to what I asked. You have defined the subset, yes or no?
Yes, we found some subsets. Yeah.
Okay. So Phil, just email Peng and he'll let you know what the he'll let you know what the subset is.
Exactly.
So check the Q&A, Peng, and chat, and and connect with the people who have questions for you.
Thank you so much. Yeah, thank you.
So, thank you. We're going to move on to our second talk. Our second talk is given by Eric Wagner, who just joined our faculty as a full professor this past summer. So we welcome him with open arms, as we do everyone in our RNA community at the University of Rochester. So he is a professor in the Department of Biochemistry and Biophysics. He's a member of the Wilmot Cancer Center and also a member of our Center for RNA Biology. Eric will talk about, there you go, unexpected interactions with the integrator complex. Thanks, Eric.
Thank you, Lynn. And I suppose I have to support that third question, eating up a little bit of my time, given it was my postdoc advisor. So I'm okay giving him a couple minutes. That's all, though, I think. Okay, so as you can see from the title of my talk, I'm going to focus on what a large portion of the lab works on, and that is the integrator complex. And I think that sort of immediately generates some questions here. And I want to spend 7-8 minutes giving some background to make this talk accessible to everybody out there. And I want to introduce 3 basic principles, some ideas about RNA pol-2 pausing, of course, what is the integrator complex, and then how we were able to sort of connect that these 2 processes had something to do with each other. And I'll use that as a sort of a platform to discuss my main story today, and that is how disruptions within interactions within the complex are actually causative of a ciliopathy disease. So in broad strokes, what we know about transcription, at least from the 1970s and the 80s, is that general transcription factors recognize elements within promoters that ultimately cause the recruitment of RNA pol-2, leading to the formation of this pre-initiation complex. Now, promoter clearance is brought about notably through phosphorylation of the C-terminal domain of RNA polymerase II. And at that point, in lower organisms, this is thought to be the rate-limiting step in order for genes to be expressed. Now, work from the 1980s and 90s and through now actually paints sort of a different picture in metazoans. And that is, as the polymerase initiates transcription at many genes, well over 50% in humans, the polymerase will then pause 40 to 60 nucleotides downstream. And that process actually is called RNA pol-2 pausing. Now, that pausing is brought by negative elongation factors. In fact, one of them is called NELF, which contains multiple subunits, as well as a more arcane named group of 2 proteins called DSIF. And together, these proteins associate with the polymerase, causing it to stall, as I mentioned, 60 bases or so downstream. And that pausing is to keep genes poised, at least that is the belief. And at some point through some type of signal transduction or response to the environment or growth factor binding, there can be various reasons for this, there will be a signal sent down to the nucleus to cause these polymerases to release from this pause state, and that then transition into elongation competency. And that is brought out by a positive transcription elongation factor, actually called PTEF-B. PTEF-B also has a kinase activity, although its phosphorylation substrates are different. It will phosphorylate within NELF, causing it to be evicted from that paused complex. It will phosphorylate within DSIF, and this is more of a nuanced event where it will change the character of DSIF from negative to positive, and it will remain associated. And it will also phosphorylate the C-terminal domain of pol-2, notably at the serine 2 position. So together, when PTEF-B is recruited, you will get essentially expression of genes and transcription through the gene body. Now, this event is not going to happen to all paused polymerases. And as we are learning now, the cell can't really afford to keep them all in the pause state. So there needs to be a process of dealing with polymerases that do not become elongation competent. And that process is termed promoter-proximal termination. Now, in many senses, this will be very similar to the termination or canonical termination that occurs at the 3' ends of genes, where the nascent RNA would need to be cleaved. But in this case, the cleavage would not need to bring about polyadenyl, but would occur in order to promote termination of that pause polymerase. And in addition, there would need to be another factor recruited in, that would have some type of phosphatase activity in order to remove those phosphates that are associated with the paused polymerase. Now, up until just the past few years, we had very sort of poor understanding of how promoter-proximal termination took place. So I want to leave that point there and sort of move to my second aspect of my background, and that is the integrator complex. And so what is integrator? It was identified in 2005 by Ramin Shikatar's lab as a group of at least 14 proteins, probably 15, that are associated with RNA pol-2, as you can see here from this IP and silver stain. Particularly the integrator associates with the C-terminal domain of pol-2. And if you inspect these subunits initially, there's no or not many predicted domains that would allow one to sort of figure out what they're doing within the complex, with the key exception of two of the subunits, integrators 9 and 11. These are paralogs of the RNA endonuclease CPSF73 and CPSF100. These are part of the cleavage and polyadenylation machinery. In fact, 73 is the cleavage in cleavage and polyadenylation. And integrator 9 and 11 form a heterodimer just as 73 and 100 does. And I'll add that my lab over the past few years has found that these two subunits associate with integrator 4 to form a heterotrimer. And that underscores that the complex itself has submodules. In fact, these three subunits are what we call the integrator cleavage module. And this module will cleave a collection of non-coding RNA, including USNRNA, enhancer transcribed RNA, and telomerase RNA. And so over the past few years, my group and others have figured out that it is not just restricted to non-coding RNA. And so this is an image from a paper we published with Ramin Shikatar's lab about 8 years ago, showing a ChIP-seq profile of integrator. And this is a metagene of the U5 snRNA genes. And you can see integrator 11 occupies these genes and is more skewed towards the 3' end, similar to the polymerase. Now, given that we observed this, and this was the result, then anything else in the ChIP-seq data I figured we could trust. And what we found is something we didn't expect, and that is integrator is not just at non-coding RNA genes, but it's also at protein coding genes, but it's not near their 3' end. It is enriched at the 5' end, in particular where RNA pol-2 pausing occurs. And so this piece of evidence, coupled with biochemical evidence from my group, showing if we pull down integrator with epitope tagged integrator 5, for example, subunit, or 11, and purify the complex and then conduct a proteomics. And these are these are peptide counts from those purifications. So you can see we pull down integrator expected, we pull down the polymerase, but we also pull down the subunits of DSIF and NELF. So this sort of indicated to us that integrator is associating with the polymerase, but it's particularly associated with paused RNA polymerase. And so over the past 3 to 4 years, my group and others have sort of unraveled a lot of what integrator is doing at these loci. And so I want to sort of summarize 2 of those papers in one slide. And so about 3 years ago, Nathan Elrod, who's a graduate student in my lab that just defended a few months ago, discovered that integrator uses its subunit 11, the endonuclease, to indeed cleave the RNA that is associated with paused RNA pol-2, and that is important to promote the termination. And then a year later, we published sort of a sequel to that paper, and Kai Huang in my lab, who was a postdoc at the time, who has now traveled up to Rochester with me, demonstrated that integrator uses its subunit 8 to associate with protein phosphatase 2A, and it uses PP2A to sort of wipe clean these phosphates that are associated with the paused polymerase. And I'll add that this work was done in collaboration with Karen Edelman's lab. Now, since these papers have come out, there's been tremendous advances in the structural biology community, and I want to shine a little light on that work. 2 labs in particular, Patrick Kramer's and the Shu Laboratory in Shanghai, have recently solved the structure of integrator associating with the paused polymerase. And I want to note that they were able to identify that the nascent RNA indeed associates with integrator 11 and gets cleaved at about the exact same distance that our work sort of calculated. And then I want to shine a little additional light on the Shoot Laboratory. They had another paper published a few months after Kai's paper, actually solving the structure of integrator associating with PP2A. And what's truly remarkable is that the key feature in Kai's paper was that the N-terminus of integrator 8 had a motif that bound to PP2A. And although I didn't label these subunits here, this light blue guy is integrator 8 and its N-terminus is kissing the subunit of PP2A. So together, I think all of these papers really put together both molecularly, biochemically, and structurally a lot of the interworkings here. And for those of you who teach classes on this sort of thing, these are a great group of papers to sort of use because there's a lot of overlapping and agreement between them. But I'm not going to talk any more about that today. Instead, I'm going to talk about some recent work that we just got into press looking at a different subunit of integrator, and that is integrator subunit 13, which we don't know much about. And so I want to tell you a story about how mutations in integrator 13 disrupt its association with the cleavage module, and that gives rise to an orofacial digital syndrome that is a form of a ciliopathy disease. So I want to back up a couple steps and sort of explain how we got onto ciliopathy disease and how we even thought about cilia as something we should be interested in. It turns out 8 or 9 years ago, we published a paper with Lori Lee's lab and Bruno Reversade, and Lori's at Vanderbilt and Bruno's at A-Star in Singapore, showing that knockdown of virtually any of the integrator subunits would disrupt ciliogenesis. And ciliogenesis, the process at which a cell builds either the primary cilia, which is in nearly all cells, and that's non-motile, or motile cilia, either one. And you can see here in these IF, if you knock down, for example, integrator 11, that cilia is not formed properly. So what is this process of ciliogenesis? Well, it's quite complex, as one would imagine. It's a very intricate organelle with many subunits and basically structural components. And if you look here, these are just example of the genes that are involved in building the axoneme or the basal body. And many things that are involved in building ciliogenesis are either direct, either because they're components of it, or they're indirectly required because they regulate gene expression of those components. But the point I want you to take here is that any disruption of this process can lead to loss of cilia function, and that can cause a ciliopathy disease. And so ciliopathies are a spectrum of disorders, probably between 30 and 40 of them. There are mutations in at least 200 genes that are thought to be causative. It is considered rare, but not ultra rare. Around 1 in 1,000 people have it. And it tends to affect multiple organ systems. That shouldn't be surprising, given that these cilia are on so many different types of cells. And this is just an example of the arrays of problems that humans can have depending on the ciliopathy that they have. Now, within the ciliopathy family, I want to drill down further to a specific subtype called orofacial digital syndromes. And there's thought to be 14 subtypes of those, and they're distinguished based upon different clinical features, although admittedly, each of these syndromes are quite overlapping. Now, there are thought to be at least now 12 causative genes that, when mutated, could give rise. Some of them are direct because they encode components of the cilia. Others are indirect. For example, this RNA helicase that may regulate gene expression. So this started in Bruno Reversade's lab, where they had 2 families that presented OFD-like 2 symptoms, which are sort of listed here. And you can see in these images, including cleft lip, polydactyly, respiratory infections, really a lot of the classic symptoms of OFD2. And so one of these families is consanguineous, so they have a relative, in the past, so they share some overlapping genomic sequences, and that could be taken advantage of in the lab by using a technique called homozygosity mapping, look for stretches of identity, and that lit up chromosome 12, and so that is likely to be a region or very high probability where a recessive mutation will be found. And through genomic sequencing and a pipeline that Bruno's lab is very good at using, and then also sequencing this other family that had this disease. And I want to point out, these families are from completely different countries, so they're not even related to each other. But in the end, we settled on and had high confidence that mutations in integrator 13 were causative of this, and these were recessive homozygous. What are these mutations? Well, they're both different because they're, again, in two different families, but they're very close to each other. They're both in exon 16, right near the stop codon. One of them changes a serine to a leucine. The other one is an insertion that causes a slightly different coding strand and then another stop codon. So this also could cause premature termination defects. And these mutations are in, oh, sorry, that one didn't quite show up on my screen. I apologize. These mutations are in a highly conserved region of integrator 13, and they're right near the C-terminus of this protein. So we looked at cells from these patients. We isolated them, primary cells. And if we look at the family that had the nonsense codon due to the frame shift, they indeed express less integrator 13. And if you add cyclohexamide, you can restore that expression. This is commonly what happens with NMD substrates. And if we look by Western, we can see a small amount of the protein being made if we have an antibody in the middle of the protein, and then none if it's the C-terminus. And then the other mutation we can see destabilizes that protein, and that can be rescued by adding in MG132. And then if we look at nasal epithelial cells, the cilia are smaller, and that is again indicative of the disease. Now, we didn't know anything about integrator 13, but using yeast-2 hybrid, we found that it indeed interacted with the cleavage module. And since we knew quite a lot about the cleavage module through biochemical means, we knew we could mutate individual amino acids in each of these subunits that wouldn't disrupt their expression, but would disrupt their interaction. And any one of those mutations, when we introduced them, disrupted the interaction with 13. So in other words, we believe integrator 13 associates with the cleavage module, but only when it forms as a heterotrimer. So we mapped the interaction motif within integrator 13. Again, we didn't know anything about the structure. It didn't have any domains that would sort of give us guidance. So this is just old-fashioned, looking at 200 amino acid pieces. And we found that the very C-terminus of integrator 13 is sufficient to interact with 4, 9, and 11. And then satisfyingly, when we introduced those two patient mutations in the context of the full-length protein, we would lose that interaction. That's shown here and again over here. Now we transitioned to mass spec and pull downs. We created stable lines expressing these two family 1 and family 2 mutations. We created stable lines and pulled down the complex, and you can see that disrupted interaction. We conducted mass spec, and we could see that almost all the subunits were disrupted, with the key exception is we didn't seem to do much to integrator 14 and only marginally affected integrator 10. And then we repeated these experiments in Drosophila by mutating the analogous region in the fly and created stable cell lines, pulled down the complex, did the mass spec, and these are heat maps, and we see sort of a similar result, that we don't see much disruption with 10 and 14, but the rest of the subunits are. That led us to test through 2 hybrid if perhaps 13 and 10 and 14 formed a complex, and we found indeed they do. And what was quite interesting is that while those family mutations disrupt 4, 9, 11 interacting with 13, they do not disrupt 10, 13, and 14 forming a complex themselves. Now, while our paper was being reviewed, another paper came out solving the structure of 13 and 14. And actually this lab, Stephanie Jonas' lab, found that indeed these 3 subunits form a heterotrimer. And what's quite amazing is that her lab was able to show that the C-terminus of 13 also interacted with the cleavage module. But unfortunately for us, that C-terminal region is not resolved in that structure. It's probably quite flexible. So at this point, we haven't been able to visualize what these mutations might do. So in the last few slides, I just want to show some of the functional data we have. We went into Xenopus and used morpholinos to downregulate the ortholog there. And what we find is indeed developmental defects similar to the humans. And we were able to stain cells for this protein right here, CCDC9, which stains cells that are multiciliated. And we can see that the cilia that are in those cells are much less profound and defined. And if we look at real fine with EM imaging, we can find that indeed they make cilia, but those cilia are just less pronounced, less of them actually, even though their structure is unperturbed. So they can build them, just not as many of them or as robustly. Finally, in human cells, we use RPE cells, which are retinal pigment epithelial cells. We knock down Integrator 13 with 2 different siRNA. We can see similar defects in primary cilia formation, visualized here or quantified here. And we did RNA-seq on these cells and found that indeed, when we knock down Integrator 13 with either siRNA, we can see a very overlapping signature of what's going on in terms of gene expression. And what's remarkable is the gene ontology really lit up that of the genes that are upregulated, again, Integrator, we think, terminates expression. So if you knock it down, you should get upregulation. Many of the genes, the first 5 categories, in fact, were involved in cilia biogenesis, and we validated many of them over here. And so I'll put my conclusions here and sort of where we intend to go with this project. So I showed you that Integrator 13 is a component of the 10-13-14 submodule, and its C-terminal domain associates with the cleavage module, and that mutations that are causative of orofacial digital syndrome 2 are also disruptive for this communication between these modules. And let's see if I can, whoop, my slides are stuck there. There we go. And then knockdown leads to aberrant ciliogenesis, surprisingly upregulation of ciliary genes, and that we can see this in multiple species. And I didn't show it here, but we've also able to do this in Drosophila. So with that, I want to acknowledge Laura Massabrota, who is basically did all the work in my lab on this as her project. She has now moved on to St. Jude's as a senior scientist. Of course, the other folks in the lab, and critically collaborators at A-STAR, including Bruno Reversade, who came to us with these mutations, although needed a lot of biochemical work to sort of figure out the molecular basis, and a mass spectrometry director, Bill Russell at UTMB. So I'll end there and I'll take any questions.
Thank you, Eric. So put your questions in the Q&A. I'll ask the first since there aren't any in the Q&A yet. What makes the genes or their transcripts regulated by either wild type INTS13 or the mutated 13 special?
Yeah, no, this is a good question. So in terms of the complex, like what recruits Integrator to certain genes and so forth, our belief is now evolving on that. I think for quite a while we thought Integrator was only at specific genes, but now I think our belief is more that Integrator is probably at most genes. And where the specificity comes into play is that it's being specifically deactivated at some genes when those genes are required to be turned on. How it gets deactivated, who deactivates it, that's our current work. But with your question about Integrator 13, that submodule is sort of a little bit enigmatic to us. The structural work that I talked about, both from Patrick's lab and the Shoot lab, that had the pause polymerase complex or the PP2A associated complex, the 10-14 module was not in those structures. Now, Stephanie Jonas' lab solved that crystal structure, but solved it on its own. So we don't have a clear understanding of how this submodule associates with Integrator. So even broad questions like, is it with all of Integrator at all of its genes, or is the 10-13-14 module only at a subset? Perhaps it's connecting enhancers to these promoters. It's kind of a black box right now, why that would be.
Thank you. So you have 2 questions, and I know I should be saying the name of the question asker, and by the way I pronounce the next one, you'll see why I haven't. This is from Sergey Shevtsov. Very refreshing to have protein-centric talks at an RNA club. Hey, it's all connected. What is a fate for non-processed mRNAs downstream of mutations, you tell us?
Oh, that's an excellent question. So it's kind of a 2-part question. So I don't know if you meant it that way, but I'm going to answer it that way. So what would be the fate of non-processed mRNAs that are subject to Integrator, mediated termination? The belief is that those are likely to be oligoadenylated by tramp complex of something of that. And then those are subject to RNA exosome decay, we have some evidence of that. In one of those papers I mentioned by Nathan Elrod, we did look at oligoadenylated small RNA sequencing when the exosome went down, and those map very well to integrator regulated genes. In terms of the mutations, what is the fate? Yeah, we don't. That's an excellent question. So what would the misprocessed or non-processed RNAs look like? Because our RNA-seq is oligo-DT enriched, we're only looking at processed RNAs there. So I can't answer your question, but I think a ribosomal depleted or some other type of more specialized sequencing would be the way to go. It's a great question for sure.
One from Bill Marslov. It seems like you should have effective integrator function at lots of paused RNAs with those mutants. Why is it so specific? Yeah, we don't know.
Yeah, no, I mean, that's sort of the $1 million question, Bill. Yeah, and I agree with you. I mean, there are tons of stories of proteins that serve general roles that when mutated give rise to amazingly specific diseases, right? I mean, and I think in many cases, we don't have great explanations why that would be. I'm totally unclear why this particular mutation could give rise to such, a unique and specific disease. We simply don't know. And I think that kind of gets back to Lynn's question of what is this 1013-14 module doing? It may be that it's playing some subspecialty role for integrator that's been evolved in later to sort of take care of some unique gene types that the core complex did not normally take care of. I'm totally speculating why that would be right now. So I'm just giving you a hand waving argument. The short answer is we don't know why there's such specificity.
Fabrice Leclerc has a question. Any cryo-EM or SAC study in progress on the integrator complex?
Well, so I did mention the papers that were out there, and I should have pointed out those were cryo-EM. So the paper by Patrick Kramer's lab and the Shu laboratory, there's actually two by them. One is in Science, and the other one is in BioArchive that I'm sure will come out somewhere. Those papers are absolutely marvelous. So, you can read those, but, in terms of more cryo-EM papers, I think there's definitely room for more of these because there's probably 3 or 4 different states of integrator that we haven't seen yet by cryo-EM. And the obvious one that I mentioned is having 10, 13, and 14 associated with it. We don't know how that submodule is in with the complex. So I'm hoping to see that at some point soon. Great question.
Okay. I have one question. It's a silly question. How come if it's cilia, why isn't it the ciliopathy? Why is it a silly? Why do you have O before?
Yeah, I don't have.
Why?
Yeah, I don't know that question. I don't know the answer to that.
Me neither. Okay. All right. Well, I want to thank everybody for participating. If you have any questions that come up after we sign off, please contact the speakers. I'm sure they're happy to answer your questions. And then our next meeting is, I don't know where it is, but you'll get an email about it. And I want to thank Eric and Peng again for a great talk. Thank you so much.
Thank you, Lynn. Thank you, everyone.